Hi, and welcome to Tom Kennedy Science, and I'm your host, Dr. Tom Kennedy. Today is part 2 of my series on the cell cycle for non-majors. Now, this really isn't about the cell cycle, but I think it's really cool and very important. I want to talk about binary fission, because when we normally talk about the cell cycle, we're talking about growth and division and the regulation of the cells in a eukaryotic cell. But I'm going to talk about the progenitors first, and that's binary fission. And I'm not going to get into a lot of details of exactly how binary fission works, but I do want to talk a little bit about prokaryotic reproduction and something else they do that's really cool. We do it too, but not as much. So binary fission. This is a form of asexual reproduction. Now, asexual, once again, A means without, and sex. So sexual reproduction means I'm going to take two genomes and merge them into a new organism. So whenever you have a plant or an animal that's creating like gametes, like sperm and egg, and you fuse a sperm and egg, well, that creates a new organism, and the organism is diploid. But in an asexually reproducing bacteria, you basically copy the DNA, move it to opposite ends of the cell, and pinch the cell in half, and you create two identical offspring, right? You only have one copy of your genome, and you're not recombining genomes to form a new organism. So that's asexual reproduction. So unlike an animal like us, you and I, we are diploid. Di means two, ploidy having to do with the chromosome number. Diploid means we have two copies of our chromosomes. So you may have heard that we've got 46 chromosomes. What that really means is we have 23 chromosomes, but they're doubled, right? We got 23 pairs of chromosomes, 23 from mom, 23 from dad, which gives us 46. And our 23 chromosomes with two copies, that's really our whole genome, and it's chopped up in all these different chromosomes. Prokaryotes are different. With a prokaryote, they've only got one chromosome, and it's circular. The ends meet. So every time a prokaryotic chromosome is replicated, guess what? The entire thing, all the DNA is copied. Now, in there with the DNA that's found in the nucleoid region, bacteria may have small amounts of plasmids, which are small circular pieces of DNA that they could have acquired from the environment. So when prokaryotes reproduce through this binary fission, which is, basically an asexual reproduction, they're copying their genes, and then they're moving them to opposite ends of the cell and then pinching the cell in half, creating identical copies of itself. Now this is an example of vertical gene transfer. And vertical gene transfer is when you have from your parents to your offspring. You're going vertical, right? So you got your genes from your parents, and that'd be an example of vertical gene transfer. There are other ways you can get your genes. Now, we often think of prokaryotes as primitive. I know, they're single-celled, they're simple compared to eukaryotic cells, which are much larger and more complex. But the fact that they can reproduce so quickly, as fast as like 20 minutes, that's a generation. In 1 hour, you can have 3 generations of E. coli in a petri dish. Isn't that fast? But what that does is that allows for a very rapid evolution of prokaryotes. And one thing they don't do is they tend not to get a large genome. They keep it small and efficient. If they have genes they're not using, they will eventually lose them rather quickly. So a prokaryotic genome stays between 4 and 6, maybe 7 or 8,000 genes, maybe a little bit fewer compared to our 20,000 genes. But by being small, including their DNA, with rapid generation times, these guys can reproduce fast, like I just said, rapid generation times, and they can evolve quickly to the environment. Now, there's something else that bacteria do all the time, and this is called horizontal gene transfer. Now, in horizontal gene transfer, you're getting genes from the environment. You're picking them up from the environment. Could you imagine if you go swimming in a lake and you got some fish DNA that's in the lake? Yes, there's fish DNA in the lake, along with all kinds of other DNA in the lake. It's called environmental DNA. We typically don't pick that up, but bacteria do. And they pick it up through what's called horizontal gene transfer. Now, horizontal gene transfer, this is not just theoretical. It happens all the time. And in fact, it's one of the reasons why bacteria can easily become resistant to our medicines like antibiotics. The reason why is because some bacteria may evolve resistance to antibiotics, reproduce, and then that DNA might escape that bacteria, get out in the environment, and gets picked up by another unrelated bacteria, and now it has resistance to that antibiotic as well. Now, the reason why bacteria can exchange genetic material like this to do this horizontal gene transfer, even if the bacteria are completely unrelated to each other, is because the DNA code of life, right, the genetic code is nearly universal. If you got a gene to make insulin in humans, you can take that gene, put it in a bacteria, guess what it will make? Insulin. Now, horizontal gene transfer in a pangenome. Let me explain this really quick. You know, the human population is about 7.7 billion people. And basically, you and the most distantly related person to you in this world are going to have 20,000 genes. We're going to have the same genes. Wait a second. I said we had the same genes. Aren't we all different? We are. You see, genes might have different versions of those genes. You could think of like the gene for blood type A, B, and O. That's 3 different versions right there. And because we have 20,000 genes with so many different variations of those genes, nobody gets the exact same combination of these various versions. And if you want to know what that's called, is an allele, is a different version of a gene. So there you have it. 7.7 billion people. We basically all have the same genes. We're just different because we have different versions of those genes. Now, of course, bacteria are different, right? If you go into your gut, you have E. coli. More than billions. You might have trillions of E. coli in your gut at any one time. I know, isn't that crazy? I mean, bacteria outnumber you like 10 to 1. I know, for every cell that's you, there's like 10 bacteria. It's just crazy. Now, when you look at an E. coli, they might have 4,000 genes. The next one over might have 6,000 genes. Whoa, they varied by 2,000 genes between E. coli. And in fact, if I took all the E. coli out of your gut, there might be like 17,000 genes in that whole pangenome of E. coli. That's almost as much as an animal. In fact, in some animals, that is 17,000 genes. So that's pretty cool that bacteria can survive as a population and have what is called a pangenome. Pan just means like all, where they are swapping genes around between each other within the whole population. That's very different than the way we do it. All humans, like I said, we have basically all the exact same genes. So I hope you find that interesting. And then here's some tidbits of information for you. I told you that, animals, we don't typically have a lot of horizontal gene transfer. But there's this virus going around right now. It's called a coronavirus. Well, viruses, some of them don't have DNA. They have RNA. And some of them have DNA. And sometimes a virus will insert its genes into your cells. And then rather than just immediately take over the functioning of your cell, they insert those genes into your genome. Wait, I know, isn't that weird? And if it gets into your gametes, then that viral genome could be passed on to the next generation. And in fact, when we sequence the entire human genome, what did we find? Bacteria genes, viral genes, plant genes, fungal genes. And in fact, it's been estimated that between 5 and 8% of our DNA came from viruses and bacteria, even plants and fungus. So, man, that really makes you think about what exactly are we, right? I know I like planting those seeds. When you start thinking like, wait a second, eukaryotes are, formed by the merger of 2 prokaryotes. So we've got this endosymbiont living within us. Maybe 2 if you count a peroxisome. We don't really know. Man, that's kind of crazy. That could have been a cellular capture there. Wait a second. Now you're telling me my DNA is like 5 to 8% of viral origins from horizontal gene transfer over millions of years? And not only that, some of those viral genes have strongly affected human evolution. And that's something I'm going to talk about when I get to a later chapter on genetics. Okay, so there's been another episode of Tom Kennedy Science, and I hope you enjoyed this one on horizontal gene transfer.